Chapter 6 is going to launch us into a brand new, exciting section of our course that deals with DNA and with inheritance and proteins and many of the things that are really essential, basic components of life. We'll learn a lot about what's controlling things in your body and what's happening in your body at this particular point in time. So we want to begin our discussion with DNA replication, but even before that, we want to have a brief review of the structure of DNA. And what genes and chromosomes, a couple other terms, what those things actually mean. So I believe we all realize that DNA is located in the nucleus of the cell, and based on our discussion class, perhaps that you knew already. And the DNA contains things called genes. Interestingly, we'll look at here in the slide of this idea, but a gene is a section of DNA that codes for a protein. And so we'll talk more about what that means in just a moment. DNA has to be arranged into some sort of structure inside the cell. And we call that structure a chromosome, or chromosomes. So the number of chromosomes varies depending on the organism that you are talking about. In bacteria, there's just one chromosome. In fruit flies, there are 8 chromosomes. Humans have 46 chromosomes. Gorillas have 48 chromosomes. The number of chromosomes varies by species, but a chromosome is simply a segment of DNA. The DNA is arranged into these long segments referred to as chromosomes. Now, here we have some pictures of human chromosomes. Humans have 46 chromosomes arranged into 23 homologous pairs. That's a term we'll come back to very quickly in our class, but right now we just want to focus on this idea of chromosomes. So on the screen we see chromosome 5, 6, and 7, and 8 of humans. And so if we look at chromosome number 5, then what we see is that the chromosome consists of a long piece of DNA. Now it takes about 23,000 genes to make a person. We have 46 chromosomes, and so those chromosomes are distributed, the genes are distributed across the chromosomes. So there are lots of chromosomes, once again, there are lots of genes found on a single chromosome. So for instance, on chromosome number 5, we have about 6 genes that are listed on the chromosome. In reality, there are thousands of genes located on each chromosome in the human body. But this particular slide is pointing out the location of the genes that code for some diseases on human chromosomes. And so fascinatingly, we can see that the gene for asthma is located right here where I'm pointing, on chromosome number 5. We know where that gene is located. The gene for epilepsy is over here on chromosome number 6. The gene for a certain type of diabetes is found here on chromosome number 6 as well. A gene associated with obesity is found here on chromosome number 7. Now, many of these things are more complicated than just one gene, but these genes have a very strong influence, for instance, on obesity or asthma or epilepsy and so forth. And so really what you are and what you're susceptible to as far as diseases go can be tied directly back to your genes, as you likely realize. So you may have heard in the past couple weeks about the situation with Angelina Jolie. She had a very radical surgery procedure done simply as the result of a gene that she carried. And so that's an example of a gene, just like the genes that we're talking about on this particular screen. She had the gene for a particular disease that is strongly associated with breast cancer. And it was just in the news 2 weeks ago that she underwent this radical surgery to prevent her from getting breast cancer. And so you should take a look at that. But that decision was made based on the fact that she had carried a particular gene, like the genes that we're looking at on this particular screen. So that's the relationship between genes and chromosomes. All of your DNA is arranged into chromosomes, and the chromosomes contain these genes. Okay, let's go on just a bit. And so here we get to the idea, another very important idea in this discussion, of sister chromatids. And so now what I'm going to tell you now is that your chromosomes, of the 46 chromosomes in your body, your chromosomes can exist in one of 2 states. They can either exist as a non-replicated chromosome, as we see on the left, or a duplicated chromosome that we see on the right. And so the difference between them is as follows. If we zoom in on that unduplicated chromosome, as has been done in the little inset there, what we see is that the unduplicated chromosome consists of one linear strand of DNA. Now it's curled around on itself. That's why that unduplicated chromosome looks so thick and short in that diagram. The duplicated chromosome is simply a a situation in which that one chromosome has been copied, and now there are two copies of that single chromosome held together at this structure that we know as the centromere. And so there's no difference at all between the DNA found on the left-hand side and the right-hand side of the duplicated chromosome. And therefore, we see that if we have gene A on the left-hand side, we also have gene A on the right-hand side, B, B, C, and C, and so forth. These two halves of the chromosome are exactly the same, and we refer to them as sister chromatids. But don't be confused, it's still one chromosome. All the chromosomes in your body exist as either an unduplicated chromosome or as a duplicated chromosome, depending on what stage of the cell cycle, which is what we're getting to later in the chapter, the cell is in at any moment in time. Alright, and so we want to briefly discuss the structure of DNA as we begin our discussion of DNA replication here. This is going to be a review. We talked about the structure of DNA back in chapter 2, and so this will be just a brief review. DNA is a double-stranded molecule made of two strands of nucleotides. The people responsible for discovering the three-dimensional structure of DNA were James Watson and Francis Crick. They were two interesting scientists. Francis Crick has since passed away. James Watson is still with us, and they came up with this idea that they were the first to realize through building some little paper and kind of stick models and so forth, and lots of actual real science as well, what DNA actually looks like. The DNA molecule looks a lot like a twisted rope ladder. So let's say we have this ladder, or even an aluminum ladder. And if we were a giant, we could take that aluminum ladder and we could twist it. And that's really what DNA looks like. The rungs of the molecule are the bases, and the so-called handrails, the sides of the ladder, are the sugar phosphate backbone. If that doesn't make sense, then look at this slide for just a moment. So what we see on this slide is that the handrails, the so-called sides of the ladder that we see here and here, are composed of sugar and phosphate groups. So this sugar is bound to this phosphate to this sugar, and so forth. And the same thing is true on the other side. And then the rungs of the ladder consist of the nucleotides that project in towards the center of this particular molecule. And so the phosphate is bound to the sugar, which is bound to the base there, and that makes up one nucleotide. So we don't want to get lost here and forget that DNA is actually a chain of nucleotides. And so one nucleotide, again, consists of a phosphate, a sugar, and a nitrogen-containing base. What I'm circling right here is a single nucleotide. This is also a single nucleotide. And so this nucleotide sits on top of this nucleotide and is connected together via a bond between the phosphate and the sugar there, which holds them together. The same thing applies to the other side of the molecule, and then the two strands of DNA are held together via these hydrogen bonds. These are hydrogen bonds that hold the two bases together. Additionally, we want to point out that there's something referred to as complementary base pairing that occurs in this molecule. This means that if the base, there's only 4 bases found in DNA: guanine, cytosine, adenine, and thymine. And so if guanine is found right here, then cytosine must be found on the other side. Its complement, as we say. If guanine is found here, cytosine is found there. If adenine was here, thymine would be on the other side. And so A and T bind together, adenine and thymine, and cytosine and guanine, C and G, bind together. So here we have a nice depiction of DNA if we zoom back just a little bit. So the red represents the sugar, the purple represents the phosphate, and the yellow in this case represents the base. That's one nucleotide, and it's bound to another nucleotide on the other side of the molecule. So you can see that DNA looks like a twisted ladder. The handrails of the ladder consist of the sugar-phosphate backbone, and the bases of the ladder that project inward towards the center make up the rungs of the ladder. Again, the rungs are held together by hydrogen bonds. Hydrogen bonds are fairly weak bonds, which can be easily separated, which is going to be important later in our discussion. When Watson and Crick discovered this structure, they said in that original paper in 1953 that this is a beautifully simple structure, and the structure indicates how this molecule can be replicated. And so they said that all you need to do to replicate this molecule is to take these two strands of DNA, pull them apart, and lay down a new strand based on the sequence of the old strand, which is exactly what happens in DNA replication. We take the DNA molecule, we pull the two strands apart, which is what's occurring right here, as I'm indicating with the pointer, and then we come along and we lay down a new strand based on the sequence of bases in the old strand. In other words, if this old base right here is a thymine, then what base would be laid down here? And you would say adenine. And if this is a guanine here, then it must be a cytosine laid down next to it. And so now we have two brand new strands that come from the parent strand, and they're both exactly the same as that parent strand.